So today we are going to hear about social bonds in Atlantic spotted dolphins. You may have already tuned in to Nicole's earlier webinar last April, I think it was. And there we learned about coefficients of association, which are one way to measure bonds or relationships in dolphins. Today she is going to expand on that. And this talk was part of, or this topic was a part of her PhD dissertation, which she just successfully defended earlier at the end of last month, actually, technically. So congratulations, Nicole, on your newly minted PhD. And with that, I will pass it over to you.
Thank you very much. And thanks for having me back to talk about my favorite subject in the whole world, social bonds in Atlantic spotted dolphins. So as Cal said, I have talked a little bit about friendships, as we casually call them, in Atlantic spotted dolphins by looking at COAs. And I will review a little bit of what I discussed in my previous webinar before I go. Put us in the right place. Our field site is Bimini, the Bahamas, which is where we study our wild dolphins. It's one of our DCP field sites. And to give you a little context, you have the southern end of Florida here, and then that funny purple color is the Gulf Stream. It's very fast, very deep water. And then the red marker shows the islands of Bimini. So if we zoom in, we can see that there are 3 Bimini islands, and then again, that purple color is showing the Gulf Stream. So you can see how close to the very deep water we actually are. And we think that the dolphins use this area and some of the shallow bank further north for all of their resting, their socializing, their playing, and all of that good stuff. And then they can easily zip off into the deep water to feed without expending as much energy as some other populations. So we have the perfect opportunity to study their social bonds. So the species, as I said, is the Atlantic spotted dolphin, and the scientific name is Stenella frontalis. Spotted dolphins can be identified based on age by their spot patterns. So they are born with no spots. For the first about month of their life, they are considered a neonate or class 1, and they have these little tiger stripes, which are the fetal folds from being folded up in their mom's belly. Those fetal folds will fade, and then they'll be considered a class 2 or a calf. We also call this two-toned because they have dark coloration on their back and light coloration on their belly. And calves are fully dependent on mom. They'll start to develop some dark spots along their belly, and then once they are independent from mom, they are considered a class 3 or a juvenile, which is also known as speckled because they have just a little speckling of dark spots along their sides and their belly. Juveniles will continue to develop dark spots, and then they will start to get some light spots on their back, and they'll become a subadult or a class 4, which is also known as mottled. This does include young adult females who become sexually mature, but males are not. And then once they have so many spots that you can't distinguish between individual spots, they are an adult or a class 5, which is also called fused because all their spots are fused together. We can identify dolphins based on, or to the sex based on their belly. There is no sexual dimorphism in Atlantic spotted dolphins where you could easily tell based on some external characteristic or their size. You can't differentiate between the sexes, so we have to see their belly. And on the left here, you can see the male has a urogenital slit, a little gap, and then the anal opening, which kind of looks like an exclamation point. Females have a urogenital slit with the mammary slits on either side, which kind of looks like a funky division symbol. And then around Bimini, we have a spotted dolphin photo ID catalog that includes at this time 117 individuals. So we identify these individuals by their unique spots and any permanent injuries or scars that they might have. So for example, these 2 photos are of number 64, Prince William, and you can kind of see that he has some scarring on his peduncle here that we were able to use to identify him before he had as many spots as he does in these photos. So dolphins are added to the catalog once they have enough recognizable spots or scars that we can identify them from one year to the next, and once we have confirmed images of both sides. So how do we collect our data? We collect surface and underwater observations around Bimini. So we do, from the boat, we'll count the number of dolphins that we see, if we're able to identify any of the individuals, but then we'll get in the water and we use a custom-built mobile video acoustic system, or MVA, which is shown here, which has a camera in an underwater housing in the center, and then we have 2 hydrophones, one on either side of the camera, and that allows us to hear stereo sound when we're listening with headphones afterwards. We also collect underwater still photos using underwater still cameras, and oftentimes it's our interns, students, and eco-tour guests who help us collect those photos. And we use those in order to identify the entire group and maximize our ability to ID the individuals in the group. Using our mobile video acoustic system, we use what we call a focal follow method. So when we get in the water, we'll pick the first random dolphin that swims into view and follow them for as long as we can. So until they swim too far away or behind an obstacle like the boat or a group of people. And we do this so that we can see every dolphin that individual interacts with, or every behavior that they present. So if they play with sargassum, or if they're investigating the seafloor or something, we're able to focus on one individual and see everything that they do. And then once that dolphin swims out of view, we pick the next random dolphin and follow them for as long as we can. This is in an effort to minimize our observer bias. So not that we have favorites, but I do. If I prefer Prince William and I am in a group with him, if I simply picked him out of the group every single time, we would have so much data about him and not as much about everybody else. So we try to minimize that type of bias, or if there's some interesting behavior that maybe we are curious about, we don't want to favor that behavior and not get a good sampling of what was happening in the water that day. So I've been talking about spotted dolphins, but this species has a lot in common with very diverse other species, including bats, spotted hyenas, African elephants, and chimpanzees. So you might ask, what do these terrestrial animals, and some that even have wings, have in common with spotted dolphins? Well, there are many things, but one of the major things, and the thing that interests me the most, is that they have very complex social systems that are managed by these really complex social bonds. And the beginning of this is that all of these species live in groups, so they have to interact with others of their own species very regularly in order to live their life. So for group living to have evolved, the benefits to the individual must outweigh the costs. Some of these potential benefits include evading or defending against predators, which includes the dilution effect. They also have an easier access to mates, because they don't have to expend energy looking for others with whom to mate. They have higher resource defense. If they have a territory where they have good access to food, they can defend against other groups of their species, or increased foraging success if you learn from other individuals in your group. They also might get decreased energetic costs if you picture penguins grouping together to retain heat, for example. But there are some potential costs, which includes increased competition for resources and mates, which can lead to aggression and therefore could be costly to an individual, and then an increased risk of pathogenic transfer. If you are spending more time very close together, if someone in your group gets sick, you have a higher likelihood of catching that sickness because you're living in a group. So how do we study such complex social systems of these grouping animals? Well, interactions are the building blocks of their social system. And these repeated interactions lead to relationships, which eventually leads to the social structure. And for example, we could, if an alien came down and wanted to know something about humans, it might station itself in like an office building. And without knowing anything about the individuals in the room, it would be able to observe for some length of time and notice the interactions between individuals. For example, if some are more friendly, if they spend more time together, if their time is spent having lunch or doing social things versus having meetings, you would be able to understand the different relationships that you have. Some might be more friendly, some might be more professional. And then by looking at all of the relationships in that office building, you would understand the social structure. You would see who is management, who might be a new intern and is learning from those above them. So that's basically what we do or hope to do with the dolphins. But for a lot of species, including dolphins, observation of interactions can be challenging. So we spend a lot of time doing what I'm doing on the boat, watching them from the surface. And as you may realize, as mammals, dolphins don't spend that much time at the surface unless they need to breathe. So we're missing a lot of the picture if we're only watching them when they're at the surface. But we can get a lot of information from association. So the time that these individuals spend in the same group can be used as a substitute for interaction. And we quantify this using a coefficient of association, or COA, which is an estimate of the proportion of time that individuals are together versus apart. It's an estimate because we can only tell if individuals are together when they're in front of us. If we see one or both dolphins, we know that they are either not together or they are. But if we don't see either, they might be together somewhere else where we're just not looking. And we only know that they're together when we are out looking as well. And it's a proportion because if we never saw two individuals together, this would be 0 versus if it was 100% of the time, we would have a coefficient of association of 1. From these COAs, we can create a network of associations. And that looks like this, where the circles would be individuals, the lines between them would indicate their association, and the thickness would show us the relative value of COA. So thinner lines would be a lower COA, thicker lines would be a higher value. So you can make a network like this in order to more easily visualize and get like a snapshot of what's happening. And it allows for insight into the social organization of the group. So, for example, our spotted dolphin COAs for the Bimini dolphins can be presented like this in a matrix, where if you found two individuals and then you found where their columns and rows overlap, you would be able to see, for example, number 69 and number 4 were seen 84% of the time together. And then you can compare that to other things. If you picked another individual, 69 and 35 were seen on average 17% of the time together. And then you can compare these things based on class, for example, age or sex. If we looked at another female, number 35 spends 41% of her time with number 80. So it's very useful to look at COAs like this and tease apart everything, but it's also a little bit easier to understand when you look at an overall network. So this is what we call a sociogram. It's the type of graph that we make from these association matrices. This sociogram is from 2003 to 2018, and it only includes pairs with the COA greater than or equal to twice the mean for the group, which was 0.1. And this was so that I could look at the stronger relation. So in this sociogram, females are shown with yellow circles and males are blue squares. And they are plotted using what we call multidimensional scaling, where individuals that are more similar are plotted more closely together and more centrally. So where you can see this little cluster in the center, the dolphins have higher COAs with their associates and therefore are more similar to each other, versus the ones that are on the periphery have less associates and they tend to have lower COAs. So what do COAs really tell us? Well, they show us what dolphins spend time together and how much time they spend together. And we're able to make some conclusions from that. So what we've learned about the Bimini spotted dolphins from COAs is that they form long-term preferential partnerships, meaning that individuals in this population will associate more with some individuals than others, and this can last over a very long time. For example, Splitjaw and Prince William, over the 16-year period, were seen together at least 70% of the time, which is a very strong partnership. So this also means they have non-random associations. It means that they choose with whom they associate or who they avoid. And we saw stronger association between same-sex pairs. And we saw that males had a higher tendency to associate in general than females, meaning that males formed relationships with others more often than females did. And because we saw stronger association with same-sex pairs, males tended to associate more with males, females tended to associate more with females. But that's not to say that we didn't see strong association between males and females. And then we also saw that males associated more with others that themselves had strong associations. But what does association tell us about their socialization? Does it really equal interaction? interaction. If we're assuming that dolphins are together in a group, are we also assuming that they're interacting, and is that a fair thing to assume? So for example, once we look at what's happening underwater, we can see some of the behaviors that occur are a little bit more complex than simply presence in the same group. This older dolphin is number 35 with her calf. We wouldn't be able to know that this type of behavior was happening. I'll play it again because it's kind of cute and silly. She's just poking at her calf with her rostrum. The COA is very valuable information, but it doesn't give us the context of what's happening when they're together in a group. So we know that social interactions are characterized by two very broad types of interaction. Affiliative ones are those that we would consider friendly. So hand-holding, shaking hands, hugging. In other species, this includes grooming and like soft body contact. Agonistic exchanges include direct aggression, like a punch to the face, but they also include subtle things like dominance and submissive behavior. So it doesn't have to be direct aggression. And this type of non-vocal communication is very important for sharing information. While humans obviously talk a lot, I'm talking to you today, but if I'm saying one thing and my body language is saying something else, we can interpret that type of behavior for each other. We're also able to interpret these non-vocal signals of other species. So for example, in this photo, the individual on the right is grooming the one on the left. The one on the left is in a very vulnerable position. So if they did not have a good relationship, they would not be doing this. So we can interpret that they have a close enough relationship that they feel comfortable for grooming. Another example is dogs. We spend so much time with dogs, we know that if we saw these two dogs, they look like they're playing. The one on the right is play bowing. It looks like it's wagging its tail. Even if we were to hear growling or see teeth bared, we would be able to interpret based on the other non-vocal signals that they are playing and not fighting. So we can do similar things with other species, including dolphins. And so we're able to study these tactile behaviors, and we know that they establish and maintain social bonds in many group living species. So we see preening between ravens that have a stronger relationship. Ravens that preen are more likely to reconcile after an aggressive exchange. For chimpanzees, grooming is, again, another form of their strong relationship. If individuals are grooming, it means that they have some level of trust. We also are able to interpret different patterns based on these tactile behaviors. Head rubbing and licking is seen in African lions, is seen between females and is thought to help strengthen the bonds that they need in order to help raise the cubs as a group. So we see this in dolphins. Types of non-vocal communication include body rubs and pectoral fin contacts, which we think are affiliative. Agonistic exchanges include bites, rams, hits, and fluke and fin slaps. And by investigating these patterns of interaction, we can uncover features of their social system. So for example, the relationship between dominance hierarchies and grooming. In chimpanzees, we see that dominant males will groom each other more often than they will groom their submissives. We see similar patterns in meerkats, whereas in brown capuchin monkeys, they'll actually groom up the hierarchy. We also see class level patterns that can be based on age or sex. So again, in African lions, females tend to lick each other more often than they lick males, whereas both males and females initiate head rubbing with males more often than they do with females. And then association-based patterns. For example, in horses, individuals who associate more are seen to physically interact more. So we use our video, and I have done this for my dissertation, to investigate tactile exchanges to try to uncover these types of patterns. And I do that by event sampling our video data, which simply means we log every time two dolphins make contact. And I'll show you an example of that in a moment. And when I find an example of a tactile exchange, I log the initiator and the receiver and include their age, sex, and individual identity if we know it. If it's an interaction where we're not able to tell who initiated, we still log the individual identity and information, but we just don't determine who was initiator and receiver. We also log what body parts they used, since we know that agonistic exchanges more often involve the rostrum and the flukes. we log those specifically so that we can later interpret what was happening. And then I also looked at individual variation and their preference for the age and sex of their partner. And I did this using some complex statistical models we don't have to talk about here. But I did want to talk to you a little bit about the results that I found, because I think they're pretty neat. So for example, when we're event sampling, if we were watching this video of these dolphins calmly swimming, we would be watching and looking for any type of physical contact exchange. For example, this dolphin on the bottom that used its head to rub against the other's belly. Another example that we see very frequently is pectoral fin contact. These two juvenile females will exchange a lot of pec fin contact, and in this we could very clearly see that the bottom one initiated with her head on the other's pec fin. And then we would have stopped our log, and here we would initiate another contact, which would be a little hard to tell who actually started it. But this is the type of behavior we're looking at when we're reviewing our video data. And then once we have a spreadsheet of all this, we're able to look at patterns of, for example, these two females. Is it juvenile females that interact more than they do with juvenile males? Do juveniles interact more than any other age class? Those types of broad questions are what we're really interested in understanding, as well as, are there preferences that might be based more on the individual than simply based on their age or sex? So what I found was that when you included calves, more contact was observed in pairs of different age but same sex, which makes sense. If calves are mostly interacting with adult or subadult females, they're mostly going to be interacting with individuals of a different age. Whereas if we exclude calves, we saw more contact between same age and same sex pairs, which is very exciting, and I'll get to that in a moment. We also saw that juvenile dolphins initiated the most contact overall, and in this figure, the initiators' age class is along the x-axis, the number of contact events is along the y, and the different colored bars show the receiver's age. So you can see that juveniles initiated the most contact, but they specifically did it with juveniles. And even more specifically, juvenile females initiated the most contact of any age-sex category. We also saw that the pectoral fin was most often used to initiate contact, and the lateral side received the most contact. So in this figure, we have the 11 different body parts that we used in our study, the number of contact events along the Y, and then the different colored bars are for the initiator or the receiver. So by looking at this tall dark bar, we see that the pectoral fin was used by the initiator the most, and then the receiver received on the lateral side the most. Once we lump everything together and we look at things that contacts initiated with a pec fin versus any body part, we do still see all body as the most common initiating body region. And then when looking at the individual preferences, in this figure, I have shown the dolphin IDs along the y-axis. These are the IDs of the initiating dolphins. Yellowish orange are the females, and the green show the males. And then along the x-axis, I have the probability that they will interact with an individual of the same or different age. So towards 1 is higher probability of same age, towards 0 would be higher probability of different age. The blue line shows the 50% mark where it would be equal probability either way, and the red line shows the overall average for this group of dolphins. So we can see that there's actually quite a lot of variation within individuals. So these horizontal lines are showing the spread, the error for each individual. So they aren't, while they do have a mean, they do also have a wide variety of types of interactions that they have shown on our videos. But then we see that there is a lot of variation when we're looking. And we saw an overall preference that was significant for initiating contact with same-aged individuals that was influenced by the individual dolphin ID. I have to look a little bit more into this, and it's one of our upcoming projects, but one of the really cool things of this is that the individual with the strongest preference towards different aged interactions is actually an adult male. And we have just casually, while we're watching from the boat and underwater, we've noticed that number 22, split jaw, spends a lot of time with individuals who are younger. So he'll either be with calves or with juveniles, whether it's playing or simply calmly swimming with the calf in the infant position as though 22 is a mom. It's very interesting that he actually shows a significant tendency towards different aged interactions. And we don't really understand what this might be about, but it could have something to do with simply he likes to hang out with younger dolphins, or maybe there's some kind of mentorship where he is helping teach the younger dolphins. So we're looking forward to teasing that apart. Some of the other individuals that spent more time with different aged dolphins are older females who have calves. So that tendency makes sense. But you can see that there are males who spent more time with different aged individuals than the overall average. So it should be pretty cool to look into that. And then we see a similar trend with when looking at same sex versus different sex. Again, the individual IDs are along the Y. The probability that they interact with same or different sex is along the X, where 1 is closer to same sex, 0 is stronger tendency for different sex. Females are in yellow and males are in green. The blue line shows the 50% probability, and the red line shows the overall group average. And you can see that even though there is an overall average, there is a lot of variability that is dependent on individual ID. And then when we're looking at the dolphins that tended towards different sex interactions, they are actually younger dolphins of both sexes. So it's possible that they haven't determined their preferences yet. They don't have an established preference for individuals of the same sex. They might also be practicing for when they get older and they're sexually mature, or they simply like to interact with individuals of a different sex. We'll have to check that out in more detail. So in summary, we saw the most contact between same-sex pairs of similar age. And this is the most interesting because it follows trends that are observed in pectoral fin contact for spotted dolphins, but also different species. And it also follows same-sex, same-age trends that we observe in other species, including for mutual social grooming between vampire bats. There are also same-sex preferences in other terrestrial species, including chimpanzees. Male chimpanzees tend to groom each other more than females groom each other, and more than males and females intergroom. And as I have mentioned before, the African lions, females will lick each other almost exclusively, whereas males and females will initiate head rubbing only with older males. So it's very exciting that the dolphins, their all body contact follows these similar trends that are observed in other grouping very social species. So with that, I'll take any questions. I've also included my email in case you think of something later or want to ask about defending a PhD or anything, or just talk about dolphins.
Thank you, Nicole. And thank you to the folks who submitted questions. Feel free to keep submitting them, and we'll see how many we have time for. Nicole, I'm going to pull out two that were kind of related, I felt. One, does mom have a stronger COA or more contact with her offspring even after the offspring is independent?
That's a great question. And it's something that I would like to look into in more detail in the future. But that actually depends a lot on the individuals, both on the mom and the calf. So we have seen some pairs where after the calf became independent, we hardly ever saw them together at all. And therefore we didn't have any recordings of them interacting. Whereas for others, including number 99, Paul, he spends lots of time in the same group as his mom, number 80, Leslie. So perhaps it has something to do with their sex. Maybe it has something to do with it, if it's mom's first calf, maybe they'll spend more time together. I can't give a specific answer. I can say that it seems to be dependent on the individual, but we don't know if there's some other.
Paul just won't move out of the basement.
Yes, exactly.
He's trying, he just won't move. Sorry if I didn't mean to derail your answer.
No, it's true.
And then in the same vein of relatedness there, do you know if genetic relatedness or lack thereof impacts the COA or the contacts? For example, you talked about split jaw and Prince William. Do you know if they're genetically related?
So we don't have genetic samples on these dolphins yet. We're hoping to get some non-invasive procedures for collecting samples soon. But based on other dolphin studies, it's actually probable that relatedness isn't as big of an issue, as big of a reason for them to either associate or interact. So you might think that if you like, you would hang out more with your siblings or your cousins than you would with others with whom you have a lower relationship. You maybe didn't grow up in the same group. But they've actually seen in other populations, including the shark-bait dolphins in Australia, that relatedness did not predict the amount of association between male dolphins, but the amount of interaction that they had as youths increased the likelihood that they would associate and form alliances as adults. So whether the relatedness might be just an extra feature of spending more time together when they're young.
And that, so some of the Shark Bay research look at like if individuals' moms were friends, like they would associate with the dolphins that their mom associated with?
Yeah, so as I had mentioned in my COA talk, one of our suggested reasons for the COAs that we observe, the strong associations, is that they grew up together and learned to trust each other and then continued to associate, and you would think that they would continue to interact to maintain those relationships so that you have a trusted partner with you, one that you know since you were young.
You touched on this a little bit, but can you talk about in the data, were the contacts all friendly or aggressive? Did you look at those differences or just the total number of contacts?
So we did look at the response of the receiving dolphin, because it's hard to interpret whether an interaction is aggressive or friendly, but you can interpret sometimes whether if an initiator approaches a dolphin and then the other one swims away, you can suggest that that is a negative reaction to that. And if it approaches and the other dolphin rubs its body against the initiator, then you can assume that that was a friendly interaction and it might be affiliative. So we look at the response of the receiver and the type of interaction. So whether it involves a rostrum or the flukes, because sometimes, like dogs, dolphins might smack each other with their flukes, but they're not actually being aggressive. They're just playing, or they poke each other around like Lil Jess, number 35, was doing with her calf. That was not an aggressive rostral behavior. So we did see overall more positive and neutral responses of the receiver than we did negative. And there was very little outright aggression overall, even in the data I looked at from 2003 to 2018.
Thank you. This next question, I'll ask it specifically to Nicole, and then if Kathleen feels like chiming in, she can. She doesn't have to. In your work that you just talked about, did you include anything about the acoustic signals from the dolphins? And then if you wanted to add anything that you are looking forward to doing in the future, or Kathleen, if you want to chime in on that at all, remind folks that Nicole did mention our DCP's mobile video acoustic system. So recording the video with simultaneous stereo audio is part of DCP's methods. So to reiterate the question, did you analyze any acoustic signals from these dolphins?
Short answer is no. While I was logging interactions, I did note whether or not there was an acoustic signal and what type, whether it was whistles or echolocation clicks. But I did not look at the relationship between those signals and the non-vocal signals. So I'll let Kathleen tackle that one.
As I unmute myself, which is appropriate for an acoustic question, we have, obviously, since we've collected the data, the acoustic data and the behavioral data, we have those data, excuse me, from Bimini to look at. And we have actually in the past had a couple of students look at some of the acoustics. One in particular looked at the type of vocal behavior just categorically associated with pec fin contact. So that was Jan Evans-Weiland, who looked at are there more clicks or whistles or a combination of clicks and whistles with pec fin contact. And that paper is available on our website, so I won't give too much of that away because I could take over this whole Q&A session. But we have looked at that, and we are looking at a variety of different questions related to the acoustics. It's just that we need more hours in the day, or more students, or more volunteers.
And also on our website is our colleague Daisy's paper on biphonation in the spotted dolphins here off Bimini. So if anyone is interested in some of the acoustic studies we have been a part of so far, make sure you check out that section of our website. We will do our little wrap up now, and then we might still have a minute or two for another question or two at the end. So if you think of anything, feel free to pop it in that chat. And in the meantime, this is a screenshot of the DCP website. And under that education tab, you can find the webinars. The first or second entry is always the upcoming schedule. So the next 2 to 3 talks that we have scheduled, and then each recording is posted here. You can also find our webinar recordings on our YouTube channel, Dolphin Communication Project, easy to find. And so we would appreciate it if you feel like subscribing to that and sharing it with your friends. And then,
oops, sorry.
This is just a reminder of our web-based educational content. Even before 2020 and COVID-19, we had our podcast, The Dolphin Pod, where hosts Justin and Laura talk with scientists, cover scientific dolphin-related topics. And we just released earlier this week our special 20th anniversary of DCP episode. So there you can hear Kathleen and I awkwardly complete an interview. I'll leave it at that. And then this was a DCP deep dive. So you can see the content is geared toward older students and adults, but we do hope that you found it accessible and interesting. And these are held generally the 2nd and 4th Thursday of the month. So our next one is Thursday, the 8th of October, also at 1 o'clock p.m. Eastern, New York, Miami time. And Dr. Justin Richard is going to talk about whale snot. So we'll get out of the dolphins for a minute and talk about belugas with him. That's next Thursday. And then our dolphin lessons are geared toward younger students, but we think they're pretty fun for grown-ups too. If you didn't catch our How to Draw a Dolphin, you can find that on the recordings. My background isn't going to allow me. That was my dolphin that I came away with. And next dolphin lesson is Tuesday the 6th, and we're going to guide young students through our website. So this will be a chance for parents to know that they can have kind of a safe website that their budding science students can learn to navigate, find some answers about dolphins, and hopefully have some fun along the way. And then I think we have one more slide. Oh, one more. And then also under the education tab on our website are kids science activities. So if you have a homeschooler, a virtual schooler, if you just like to continue learning, you can download. Those are all PDFs for free. They're anything from activities to do to guide kids through to word searches and coloring pages. So lots of fun there. And now I think it's the last slide. So we've covered lots of questions. We hope you enjoyed this talk. A reminder where you can find us, how you can stay in touch by email or social media. And then of course, we wouldn't be a good nonprofit if we didn't give a plug for how you can support us. We are happy to provide these webinars for free and keep them accessible to everyone. And we do rely on our supporters support for that. So you can adopt a dolphin, you can become a member or make a donation. We have t-shirts, we have kind of affiliations there like Amazon Smile. And of course, when it's safe to travel, you can join us on an eco tour. So thank you again. And a big thank you to Dr. Nicole for guiding us through that and sharing more of her research with us. I never get sick of hearing about it.
Thank you.
Thank you, and thank you, everyone.